Hello, dear students. I welcome you all on behalf of Sri Jendra Rangjon classes. Dear student, we are in a continuation of the chapter that is plant growth and development. And in this, we are in a continuation of the topic, one of the important topic that is plant growth regulator. And in that, we have already discussed about the auxin, gibberellin, cytokinin, and ethylene. Today we are going to talk about the one important regulator that is abscisic acid, which is actually not a promoter, it is a plant growth inhibitor. Okay, so we can write it as a plant growth inhibitor, and this is also known as stress hormone because it is getting synthesized in the stressful conditions and like a drought, waterlogging, and the adverse environmental condition in that the synthesis of this particular stress hormone take place. Because of this, it is known as a stress hormone. Now, so abscisic acid, it is known that during mid 90s, 1960s, we can say, three independent researchers reported the purification and chemical characterization of three different type of inhibitors which are known as inhibitor B, or we can call it as an inhibitor beta, then abscin 2 and dormin. Okay, these three different inhibitors was discovered by three independent scientists. Now, later on, it is known that all these three were proved to be chemically identical. So it was named as or grouped under a name that is abscisic acid. Okay, so they all are the particularly a same one. Now, here, if we talk about this inhibitor, name of inhibitor, name of the researcher, and the source from where this particular inhibitor was synthesized, that if you want to know, then first is the obviously we have understood just that is inhibitor B. It is known that this inhibitor B was discovered or researched by Bennett Clark and Cafford, and that is specifically in dormant potato tubers. So this was the first chemical discovered from the dormant potato tuber, that is inhibitor beta. The second inhibitor is abscission abscissin 2. This abscissin 2 was discovered by Endicott and Okuma, and this was from the source that is mature cotton fruit. Mature cotton fruits. And the third is dormin. This dormin was researched by Waring and Robinson. Waring and Robinson from the old betula leaves. So this was the three scientists who discovered three particular different chemical inhibitors. But later on, as we have discussed, all three were proved to be chemically identical. So they were named as abscisic acid. Okay. So this is what a particularly we can call about the discovery. Now, obviously, as we had discussed, it is also known as dormant because it induces the dormancy of bud and ground stem and seeds. So, if we talk about abscisic acid, it is considered that it is mildly acidic. It is mildly acidic, dextrorotatory, dextrorotatory, cis, cis-squai terpene growth hormone. So no need to remember this much difficult name. Just you if you remember that it's acidic hormone, then it is okay for this. And it is obviously inhibitor. There is a counteracting effect, has a counteracting effect compared to the other oxygen gibberellin and cytokine. First isolated by Edicott et al. In 1963 from cotton balls. Okay, so this is the what we can say about the particular discovery. Now, if we talk about the synthesis, then precursor is carotenoid. Is carotenoid, that is viola xanthin. Okay, so it is known that this viola xanthin undergo oxidation and form abscisic acid. Okay, so this is the thing. So abscisic acid plays an important role in regulating abscission. So if we talk about the function, obviously it plays an important role in regulating abscission and dormancy. So during abscission, there is a falling of a leaf, or we can say flowers, fruits. The abscission layer, a separation layer, is formed. It acts as a general plant growth inhibitor and inhibitor of the plant metabolism. Obviously, ABA inhibits its germination as well as because of this, it is a seed dormancy carrying out hormone and dormancy of bud specifically it causes as it approaches the winter. So apart from this, ABA is also known to show the carry out a closing of stomata in the epidermis, stomata which are present in epidermis. So in most situation, ABA act as a antagonistic to gibberellic acid because we know that gibberellic acid carry out germination of seed and ABA carry out a dormancy or inhibition of seed germination. Closing of stomata is also carried out by the ABA. Increase the tolerance, it is known as stress hormone because it increase the tolerance of a plant to various kind of stresses, and during the stressful condition its synthesis take place. Because of this we call it as what, stress hormone. Apart from this, if we talk and if we discuss some more important points in the detail one, then first it cause seed dormancy. So the question is why it causing a seed dormancy, because dormancy allows seed to dormancy allows seed to tolerate desiccation and and extreme of temperature. Desiccation and extreme of temperature. Okay. And bird as well as seed, it is known that bird as well as seed sprout only when sprout only when abscisic acid is overcome by gibberellin. So if the concentration of gibberellin increases in the seed, then it will cause a germination. So because of the action of inducing dormancy, abscisic acid is known as dorming. Apart from this, it is known that abscisic acid stops abscisic acid stops mitosis in vascular cambium in vascular cambium towards the approach of winter. Apart from this, if we talk about some more functions of abscisic acid, it is known that excessive presence of ABA stops protein and RNA synthesis in leaves and hence stimulate their senescence. Actually, leaf roll is promoted by ethylene, but leaf abscission is also caused in excessive presence. Apart from this, as we have discussed, it closes a stomata, so it prevent transpiration by closing stomata. Then ABA inhibit, actually germination is why taking place in the seed, because when the gibberellic acid gets synthesized, it's carrying out reserve food mobilization and that lead to the germination of the seed. But what ABA will do? Abscisic acid inhibit gibberellin mediated amylase formation during germination of cereal grains. During germination of cereal grains. Okay, so that is ABA. After this, in small quantities, ABA promote flowering in some short day plant. Some short day plants. Example, strawberry and blackcurrant. So in that it promotes flowering. Apart from this, if we talk about some more function, ABA found to induce parthenocarpic development in rose. So in rose, parthenocarpic development was induced by this ABA. Then next, root formation of rooting in stem cutting is promoted by abscisic acid. Example, bean, ivy, poinsettia, that is euphorbia pulcherrima. In this, the rooting in stem cutting is promoted. Then this ABA is antagonistic to gibberellic acid and counteract the effect of other growth promoting hormone, that is auxin and cytokinin. Therefore, keep their activity under check. So, the activity of auxin and gibberellin are not shown in the presence of this particular abscisic acid. Apart from this is the important statement of NCRT, which usually they ask that ABA or abscisic acid plays an important role in number one, seed development, and number two, that is seed maturation. So these are the two important things in which ABA play important role. Normally it inhibits seed germination, okay, but it has some important event in these two things. Okay, so that is what we can say, and it reduces transpiration. Because of this, we call it as what, anti-transparent. So that is all about what we can say, the abscisic acid. Okay, I hope, dear students, you have understood this about abscisic acid discovery and the detailed functions which are obviously known or shown by this abscisic acid. I hope you understood well. Thank you.